I think there's two big uses from kind of a clinical application here. One is it's going to create big changes in the drug development market. So I'll address that one first. But then I want to talk about how it's going to change diagnostics, right? Which of course meshes with drug development and drug treatments, but it's going to have big impacts there as well.
Hey, smart scientists. Welcome to another episode on the Smart Biotech Scientist podcast. Would you like to know more about the latest innovations in next-generation sequencing? Well, you are in for a treat today because I'm having a conversation with Jonathon Hill, who is the Vice President of Science and Technology and the co-founder of Wasatch BioLabs and an Associate Professor of Cell Biology and Physiology at Brigham Young University. And we'll be talking about innovating genetic sequencing. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Jonathon, welcome to the Smart Biotech Scientist podcast.
Thanks. It's awesome to be here. Thanks for having me on.
Yeah, it's a pleasure to have you on the show today. Jonathon, share something that most people don't know about you.
Most people don't know about me, huh? So I have strong connections to Brazil. In fact, last year, I went down to Brazil on a Fulbright scholarship, which is kind of a foreign exchange program for scientists. And I taught a bioinformatics class and sequenced the genome of a species of sponge that they have down there. So I had to learn how to scuba dive and got to go in the ocean, do a little field research. It was a really great experience, and I made some really great connections down there.
So it's pretty fun being a scientist, isn't it?
It is. It's fun that we get to do, kind of explore what we're curious in and do what we want to do. And I have great control over that and being able to decide what questions I want to pursue and what research I want to conduct. And I get to just be curious every day and discover something new regularly.
How did you get started in biotech, and what pivotal moments and decisions brought you to your current role?
To really get there, I kind of have to go back a ways. Sometimes I feel like an old grandpa telling stories from my youth. But when I was an undergraduate in university, I was pre-med, like most people. But my motivation for that was I knew I was good at science, and I knew that I wanted to have a career that would pay well, but mostly that it would help other people. And growing up, that's what I saw doctors did, was they got to help other people every day using their kind of scientific knowledge and things like that. And so I was doing that. And as I started going through undergrad, I started doing research in a lab and TAing a class, a genetics class. And as I did that, I started realizing, wait, I'm helping people every day. And I love doing the research and I love teaching these students. And so I changed my career path. I wanted to go a different way. So I went to graduate school instead of medical school, ended up going to Columbia University in New York, had a great experience there doing research, and then kind of did a postdoc and got a faculty position, kind of the typical route there. But then as I got into the faculty position, I started realizing that I had gotten trapped in this kind of grant publication cycle that people were talking about, where I'm trying to do research, not because I'm super curious in it, but because it's what I can get grant funding for. And then I do that to get a publication because that's going to form the basis of my next grant. And I got kind of into that cycle. And I started thinking, I did this because I wanted to be able to help people with my research. And we were discovering cool things in the lab. And I don't want to say that that kind of basic fundamental research has huge value for increasing our knowledge as a whole, and everything we do is based on what was discovered there. But I started thinking, is there a way that I personally can start making sure that my research is impacting people? I first started getting into technique development, thinking, can I help scientists gain new tools to be able to do things that they couldn't do before, answer questions they couldn't answer before? And I had some great students that came up with some great ideas. And we started thinking, okay, how do we get this tool out there? How do we make sure that scientists have access to it and can be able to do it? And the answer was we had to go through the IP process and create our own company and do it. And so my first company was in conjunction with a student. I was co-founders with him. He had had a great idea for a CRISPR-based tool. He was able to develop it in the lab, and then we created a company for him to take, and he's CEO of that company and is running with it. And I think because of that, this latest one, what happened was another student in another lab had a great idea that he started to develop. And it required nanopore sequencing, which here at my university, I'm kind of the nanopore guy. And so they came to me and they said, hey, we know you're interested in this commercialization. We know you're interested in impacting people's lives. Do you want to be part of this? And so they invited me to come in. I was very happy to do it. And we've really worked hard to think, how can we develop this technology so that it can impact people's lives, be accessible to people, and really make a difference out there? And so that's kind of how I got into this. And it's been a fun journey. I've realized that I can know a whole bunch about science and not a whole lot about business. And I've had to learn this whole new skill set and vocabulary and rules and all of these different things in order to take that next step of moving the great research that we have in academic labs into production, into the public sphere where it can really impact people's lives.
Wow, that's fascinating. I love that you are not only driven by your passion, but it looks like you have a purpose, you see a purpose, and that this gives some significance. And I love that. It makes what you're doing all more powerful if you have a purpose. Let's talk about the next generation sequencing, because there's a lot going on in that space. Can you tell us about the exciting advancements in this space?
Yeah, so next generation sequencing has for a long time been a super powerful tool that we can have. It's kind of followed me throughout my career. When I was just starting university was when the human genome was published. And so I remember seeing that, and I remember seeing everybody talking about how having that reference genome was going to change science and change how we do things. And in many ways, as I've gone through my career, that's true. The way we ask questions, the kinds of questions we can ask, and our ability to explore things in new, unique ways has fundamentally changed since that publication. When I was in graduate school, I started working with Illumina sequencing, so 2005 to 2010 kind of range. And it was amazing to me how instead of doing this kind of guess and check kind of system where we have to say, well, I think maybe this gene is involved, and then go see if we're right, we could just say, well, what genes are involved, and go explore them all and measure them all at once and be able to look at things simultaneously. At the time, and even now, I do a lot of exploration into gene regulatory network data analysis type things. And so the fact that we can actually look at this as a network now has really changed our research. But it wasn't done. I mean, you first had that kind of short read sequencing that was huge to allow us to do things and still has a lot of utility today. But over the last 10 years or so, we've started to see new technologies come out that allow for longer reads. And this gives us better resolution in the genome. We can explore the repetitive regions. We can create better assemblies of the genome. When I did my research as a Fulbright scholar, that was hugely important to be able to assemble a genome faster and more easily than we ever could before. We did the entire thing in about 4 months. And that is amazing that we can take a genome from nothing to completely assembled in less than a year. And on top of that, I think the next wave that we're starting to see is some of these sequencing technologies are now allowing us to look at methylation marks and other epigenetic marks and chemical modifications to the DNA. And this has been a world that we've known about for some time. We knew that the DNA got methylated, but we just didn't have the technology to really explore what those methylations mean and how they can be used as biomarkers for diagnostics and different applications. And now with the technology and its rapid pace of improvement, we can explore these questions and we can look into this and get a deeper insight into biology. And just to illustrate the importance of this, one of my main foci is looking, like I said, gene regulatory networks, but specifically in heart development, how an embryo gets its heart. And when we got the Human Genome Project, a lot of people thought, okay, we're going to understand all congenital heart disease at this point because it's a genetic disease and we're going to find the mutations and that's going to be it. Well, as of about 2018, the last time that it's been published, 35% of congenital heart disease incidences can be explained by a genetic mutation. So that means almost 2/3 of cases don't have a mutation that's causing it. And so now we're saying, well, what is causing it? Are there environmental factors that are interacting with things? And are there epigenetic changes that are resulting from these environmental factors that are changing gene regulation in some way that's disrupting heart development? And so I think we're going to gain an even deeper insight to these diseases and how they function and what creates them by diving into the epigenome and looking at these things that we've never had access to before.
Wow, that's exciting. I mean, and this is just in a few years. It's really powerful. I'm wondering, how will these technological advancements actually revolutionize the development of drugs in the future? I mean, can you give us an overview on that?
I think there's two big uses from kind of a clinical application here. One is it's going to create big changes in the drug development market. So I'll address that one first. But then I want to talk about how it's going to change diagnostics, right? Which of course meshes with drug development and drug treatments, but it's going to have big impacts there as well. On the drug development side, first and most obviously, it's going to create new targets. I love kind of the imagination and the creativity of students. And I just had a student the other day coming to me and saying, well, couldn't there be a treatment where we take Cas9 and fuse it to a methyltransferase and just change the epigenetics of the disease? And that's just a drug paradigm that doesn't exist right now, right? Messing with the epigenome and changing things that way. I think there's going to be drug susceptibility or effectiveness, those kinds of things that are going to be tied to epigenetic states in the genome as well. We have many drugs that work most of the time, but every so often hits a case where it just is not effective. And some of those are DNA mutations that cause that, but others are going to be gene regulatory pathways and things like that. And now we can explore that and look into that deeper. You think about cancer drug development specifically, and on both sides, you can see that, right? Where deciding if a cancer therapeutic is going to be effective in a particular case will depend on not just the genetics, but the epigenetics of that cancer tissue. And then also on the other side, perhaps there's some targets there where we can try to shut down an oncogene by changing the methylation state or something like that to help affect and treat that cancer. Now, on the other side, another important thing that perhaps isn't as well thought about out there is that these epigenetic marks become hugely valuable biomarkers for us to look at in diagnosing the disease. For example, I love talking to my students about how every cell in your body has the same DNA, but a cell in your eye is very different than a cell in your toe. And so what's different is the gene expression profiles that these cells make, which determines the proteins that they have, which determines their shape and function. But what controls the gene expression profile are these epigenetic marks. And in the case of DNA methylation, these are marks that are on the DNA, and they change through your life. They change with environmental factors. They change with disease states, all of these different factors. And we can use these biomarkers to look at the state of disease. And one simple application that we've been exploring is that you have DNA floating around in your blood. It's not any cell or anything like that. It's been released by dying cells, and now it's floating around in your blood. Well, we can access that, take a liquid biopsy, take a blood draw, and look at the methylation states of that DNA at particular sequences and determine where that cell-free DNA came from, which tells us which cell type is dying in that particular disease. So you can think about neurodegenerative diseases where, hey, if a certain cell population is dying, we can identify that by the unique methylation marks that only exist in that cell type that are now found in the cell-free DNA in the blood and be able to diagnose that disease in a minimally invasive, rapid, and accurate way. Other diseases, autoimmune diseases or different types where you have some sort of cell death, all become available through this simple biomarker.
Hey, smart biotech scientists! Have you been enjoying our conversation with Jonathon Hill? Jonathon Hill is the VP of Science and Technology and the co-founder of Wasatch BioLabs, and he's also an associate professor of cell biology and physiology at Brigham Young University. We are continuing our conversation on next generation sequencing on Thursday. What do you think of this episode? Please tell us by leaving a review on the platform you found us on. And perhaps you are wondering how you can wire in novel technologies in your development workflow. If this is the case, I'd like to offer you a free clarity call to help you get started. Use the link down in the show note and I'll see you on the call. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.